I'm based at Canada's Michael Smith Genome Sciences Centre in the group of Dr. Stephen Jones. And we've been running, as Anna said, we're part of the Provincial Cancer Agency, and we've been running a very successful personalised oncogenomics clinical trial for the last 12 years or so, using short-read sequencing, but we have short-read whole genome and transcriptomes from just over 1,000 cases. And in about 83% of those cases, we can find clinically actionable findings, often with accompanying targeted therapies. In just under 40%, we actually are able to administer therapies. In our trial, we are typically recruiting people with few other lines of therapies with fairly advanced cancers. So a lot of the reason we don't end up giving therapies is that people actually pass away or are too ill to be receiving further therapies. So we've been quite successful, I think, in improving patient outcomes through this. But we've been very interested in the potential for nanopore sequencing to improve upon what we're getting out of the Illumina. And so we've sequenced a cohort of 186 diverse tumors on the PromethION at the beginning of last year. This is all R9 chemistry because the Kit 14 hadn't quite come out yet. 39 of these with matching normal, and we ran this all through using Hamilton Nimbus 96 to do all of the automated preparation. We didn't do any shearing, G-tube shearing, which I'll call out just in the next slide. Sequence it all on our PromethION. And I've just listed all the various software we've done. We've been primarily interested in structural variant calling, DNA methylation, and phasing. So this is just a summary of the data we've generated, averaging about 70 gigabases per flow cell. Got a good 17 terabases out of it. And our N50s are actually very good, averaging about 30 read N50s, 30 kb or so, right up to about 40 kb. So the first thing we were kind of looking at was somatic structural variant detection. This can be quite difficult to benchmark, but one of the ways we benchmark it is on this Colo 829 cell line, which is fairly well characterized as a cancer kind of ground truth. And from nanopore sequencing, we were able to detect 39 out of 39 somatic ground truth structural variants. Using an ensemble approach of 3 different structural variant callers, none of these structural variant callers could actually get every single variant consistently. So we found, as in the Illumina space, that an ensemble approach was the best one. And then we went in and selected a short list of very well characterized recurrence clinically relevant structural variants from our cohorts, and just checked that Nanopore was able to detect these, and all 7 of these that we selected were able to be detected, notably from only about 20 to 30x coverage, whereas Illumina we're targeting close to 100x coverage. So we were actually quite pleased at even giving tumor content and other limitations that we were able to detect all of these. And then kind of obligatory, here is an example of a complex structural variant which we could detect in the nanopore. This is in a cancer-relevant gene in SMG1. Quite complex, there's about 4 or 5 breakpoints. We saw maybe 3 reads in the Illumina to support any of these breakpoints. It was completely invisible to Illumina sequencing. So this is just a kind of example of where, as was kind of shown by Helen this morning, you have these structural variants that are completely undetectable by short-read sequencing. And this was detected with just 28x coverage, even though it's a relatively kind of subclonal event. DNA methylation is also what we've been very interested in, and on the left you have the obligatory correlation plot. One of these samples that we sequenced had whole genome bisulfite, and we get quite good correlation with that. We used nanopolish because that at the time was really the state of the art. We're moving much more towards using Remora as our standard. And then just to kind of show, yeah, this isn't the updated plot, but anyway. Just to show a rough kind of general trend that you might expect when you take some comparable normal tissues from, it wasn't actually from GTEx, it was from IHEC, and you compare our tumors, that you see kind of like difference, lowered methylation in the tumors, and like we see the opposite in CPG islands. So that's not a novel or huge result, but it's just reassuring that we're seeing what other people have seen in cancer cohorts when they've done deep dives into methylation. But something we are a little bit interested in, this is 2 t-SNE plots of the same set of tumors. And they've been separated here based upon enhancers, CDCF sites, and promoters as the kind of distance matrix. And what we see in our cohort, we have a lot of metastatic cancers, and it's not always clear what the tissue of origin is of the tumor. So it's quite important if we can be able to ascertain the tissue of origin to help guide therapy. We usually do this right now through looking at the transcriptome and comparing to known other samples, but this really kind of indicates that the methylation clusters by tumor type and not by biopsy sites, and we're quite excited about the possibility of using this as an orthogonal approach for detecting tissue of origin. And then phasing is kind of the other really quite exciting potential for Nanopore in this sort of space. This is just kind of looking at a very rough assessment of phasing. That first plot on the left actually says quite an interesting result in that, you know, to get into the kind of megabase-sized phasing blocks, you really do want a read N50 of probably about 20 kb or more. And that's also quite interesting when you compare to the other long-read sequencing technology, which does tend to cap out at about 20 kb. And then the other two plots are just showing coverage of genes, and we've actually highlighted in the far right tumor suppressor genes, which I'll get to in the next slide as to why we're interested in them. And we can phase most genes, capture the entire gene body in one phase block in about 85% of genes across our cohort, which is quite exciting. And even some of these enormous genes, you know, that are kind of megabases in size, we can capture them, you know, 25 to 30% of the time, which is the entire gene body, which is quite impressive. So why this matters, there's this thing called the two-headed hypothesis, which I think most people are familiar with, which says that a tumor suppressor gene needs to be inactivated on both alleles in order to drive cancer. In POG, if we see two small variants on a tumor suppressor gene, we assume that they're entranced to each other and that they're inactivating it. But with the long-read phasing, we can actually go and investigate this. And in fact, this image is showing a case where the two short variants were actually in cis to each other. This tumor suppressor was actually knocked out on the other allele, but by loss of heterozygosity, which you can kind of see with the blue reads on the other haplotype being lower copy number. But that was a case where we would have assumed that it was due to the small variants and not from the copy number. And it means that if we do have a case where a tumor suppressor hasn't been knocked out, that both small variants are in cis, we can actually detect that. And this is just, Katie looked at all sort of 27 cases we had. In about 67% of cases, she could reliably tell whether they're in cis or in trans. And in the other cases, we had good reasons to not be sure. And then the last thing for phasing is that we're able to phase methylation as well as take our short read transcriptomes and phase that and link it up to the phasing information. Mostly what we found with that is a lot of allelic expression is actually explained by imbalanced copy number. But there is some that's driven by differences in allele-specific methylation, and there are some nice examples, which I don't have today, but we will have in the upcoming paper, where we look at promoter methylation that's inactivating one allele of important tumor suppressed genes. So just to summarize, we've sequenced 186 tumor samples, 39 with matched normal, from 178 advanced cancer cases, some of them we had multiple biopsies. Phasing works very well at finding multi-hit TSGs. And we can recall clinically relevant fusions. We can also detect structural variants that aren't otherwise identifiable from short-read data. And our plan is to shortly release this data via EGA and any of the fully anonymous data through a slightly more direct method, and to try and get a paper out shortly so we can provide this as a resource for other developers of long-read cancer analysis software.